back to DeSci.Party. So today we are live with Dr. Jocelynn Pearl. I myself am up at ETH Global, the hackathon in SF. So on the floor here live. And we're going to be talking about DeSci. We're going to be talking about all sorts of fun things that Jocelyn's working on. So welcome, Jocelynn. Thank you for joining us.
Thanks for having me. I'm excited for the DeSci.Party.
Right. Yeah. Science is supposed to be fun, right? I always used to say, well, now I say all the time because my advisor used to say, you'd walk into the lab and we're just like heads down working, completely quiet. You'd walk in like, everybody having fun? Yeah. We're like, yes, of course. Like, I'm just pipetting away.
Oh my God. Yeah, one of the first labs I was in at this biotech, the labs were split up into individual rooms. And so it was a little bit easier to like play music if it was just a few of you in there and you all agreed on what was going to play. So we called our lab Club 1. That was, some late nights in there listening to music together. So
right.
It's good to have fun.
Different rooms, different parties.
Yep.
Nice.
So how's the hackathon going? Like you were at DeSci Day yesterday, right?
Yeah, so there was a DeSci focused event. It was held, it was a side event, it's an official ETH Global thing, but it was all DeSci SF, hosted by WeaveChain. So shout out to Omar who put that together, did a lot of work. It was a great event. Protocol Labs and Frontier were the keynotes. So, not Frontier,
Foresight, yeah.
Yes. Yep.
Was Allison there?
Yep. So she gave the talk.
Nice, that's awesome.
Yeah, it was
very cool.
So I met a bunch of DeSci folks. Went out afterwards. We had our DeSci happy hour. It was fun.
Nice, that's awesome. Yeah. What were some of the highlights?
Well, there was I think, obviously, Foresight is doing amazing work. Their tech trees have just come so far. Allison did a little demo of that. That was super cool. Let's see, Space DAO was awesome. So working on not just advancing space, humans in space, but also longevity. Really, they're studying the intersection of how basically being in space reduces your lifespan, and it basically has this. They're using like low gravity as a model for aging. It's really interesting, and database that was really cool too.
Because you age faster in space, right?
Yeah, way faster.
Yeah, I forget the name of the astronaut, but there's a well-known astronaut who's a twin, and so they did like a twin study essentially tracking like a pretty cool like personalized medicine or quantified tracking study of all the changes, comparing the two of them.
Mm-hmm.
It's interesting.
Takes years off your life to be an astronaut.
Yeah, and it like completely changes your immunology too, right? I don't know. I haven't been in that area as much since grad school, but pretty exciting work. So.
Yeah, definitely. Yeah, they've done like multi-omic studies of comparing, actual aging versus being in space, and it looks like most of the markers line up. There was a small subset, I think, that they said that didn't, but they seemed like it was a pretty good model.
Mm-hmm. Cool.
Yeah. So let's talk a little bit more about your work. So you are a senior scientist at Tune Therapeutics, also host of the podcast, the Lady Scientist podcast. So go check that out, anybody listening. Fantastic content. And so you interviewed just female scientists on that show, right? Just women scientists?
Yeah, I started it because I just saw all of these podcasts that only interviewed men, and so it's a bit of a reaction to that. But there's just so many awesome women scientists out there doing incredible work that I just didn't see featured in media. So I just did an interview actually an hour before this with a basic researcher from Fred Hutch. And yeah, I love talking with women and featuring their career journeys and helping to inspire the next generation. And maybe one day there'll be more women focused podcasts, or I'll see more of these awesome women represented in media, and I won't have to keep only interviewing women. I love chatting with everybody. So I don't mean to be, only focused on women all the time, but
Yeah. Well, no, we need it. I mean, science in general is such a male-dominated field, right? And so traditionally is tech and Web3. And I think that's something I really appreciate about the Web3 space is that there is, active awareness and, intent to be diverse and be inclusive. And events like this and women-led events, things like that aren't necessarily exclusive, right? Men are welcome to support and come attend and learn. So I always like to highlight that as well.
Yeah, for sure. Yeah, and I think one of the exciting things about Web3 and some of these online communities is that hopefully the bar for access is a little bit lower, and anybody who's interested in being involved in some of these spaces or these topics can get involved, and it be as easy as clicking into a Discord server, and you can start chatting with other people interested in the same thing. So it's a pretty exciting aspect of the space.
Right. Yeah, it's awesome how like that communication aspect, and it's not even really Web3. It kind of happened with like open source software and people coming in and having that community, the direct connection to the quote unquote developer, right? If you're a user of science, having a connection to scientists. I say everybody is a scientist, but ultimately, people practicing doing science with their data, right, versus people who just sort of consume it. Bridging that gap is a huge thing that I see DeSci doing, even though it's not necessarily even Web3 feature. We're just communicating. I'm just
Yeah, I like the quote. I repeat this all the time, but from Cabin DAO's founder about like embracing the principles of Web3 and this like open source, permissionlessness, shared ownership. Some of these conversations or themes or principles have been really valuable with regards to science, I think, because so much of like our current scientific system happens in these ivory towers, so to speak. And so I think all of those discussions are valuable. It doesn't necessarily have to happen on the blockchain, and certainly a lot of the projects and communities that I'm a fan of don't use any blockchain technology at all, but they embrace, this theme of decentralization, of bringing in people from all different areas. There's often like globalized teams that are working on problems. And yeah, I think it's all really inspiring that there could be a better system of us working together to solve real problems or human suffering.
It's all coordination all the way down.
Yeah.
Right. I love the down means. But cool. So let's talk about a few things. I've got a bunch on the list today. But let's maybe talk more about your background as a scientist. How did you get into science, and then we'll get into eventually what you do at Tune.
Yeah, I was always very interested in science and biology as a kid, and life, animals. I thought I wanted to do outdoor science for a while, which is my nickname for things like ecology and animal behavior. But grew to really fall in love with genetics as an undergrad at UC Berkeley, and kind of changed gears and started working at the bench, studying genes and different diseases, and fell in love with biomedical research, essentially. So working on drug development for different diseases, and did that, and then went on to get my PhD at the University of Washington, where I studied gene regulation in different brain diseases using computational and experimental approaches, and then went back to biotech. So back to working on drug development, which is one of my passions.
Awesome. Yeah, I can get with the love for genetics. It's also my background as well.
Nice.
So I studied molecular cell and developmental for my PhD.
Awesome.
Yeah.
What like specific area?
I worked on, so I went to UCSB for grad school, and I worked on marine invertebrates. So we were right on the coast there, so it was great. We went down to the harbor, straight them off the side of the dock, or like the bottom of the kayak or something. They were tunicates, Cystidians. So
Okay.
the rest of the lab studied neural development because we could get thousands of embryos and just work with them in seawater at room temp. And they're completely translucent, so you could basically do imaging studies on neural development, watch the neural plate form, watch the cells move around as they differentiate, and so that was super cool.
Wow, that's so neat. I didn't even know people were studying neural development in that type of organism. That's really cool.
Yeah, they're phylogenetically at a really key point on the tree. They are invertebrates, but they're the closest related to vertebrates, right?
Okay.
The Ascidians and tunicates. So in terms of being able to practically in a lab as a model organism, they're really flexible because you don't have to get approval for vertebrate protocols and things like that. And again, thousands of transparent, translucent embryos to work with in a dish.
That's very cool.
Yeah. And there's
Have you jumped in? Oh, go ahead. Sorry, I missed that.
Sorry, I was going to say, and they develop pretty synchronously. Kind of like that's an advantage of like Drosophila or C. elegans or something like that. Is that especially C. elegans is that really synchronous, reproducible. So sorry, I didn't mean to cut you off.
No, I was just curious if you'd heard of New Atlantis DAO and if you've hopped into their server at all. They're working on a pretty cool like metagenomics pipeline for ocean water and have some exciting research going on.
Definitely, yeah, I love New Atlantis. It's great vibes too. Like it's just a fun sort of dystopian.
Yes, totally.
Yeah. But yeah, so that background is in genetics as well. And so and you're working in drug development at Tune. And so what brought you there? What attracted you to that? And then I don't know how much you can talk about what you're actually doing, but I would love to hear about that as well.
Yeah, so I think a lot of folks are familiar with like gene editing technology, basically CRISPR, and there's actually a couple other modalities for gene editing that I worked on before I went to grad school. So zinc finger nucleases and talons were both, kind of predecessors to CRISPR in a way, as something we could engineer to target a specific part of the genome and induce editing. So that system was really exciting for people who thought that we could use that to cure genetic disorders. But it's actually somewhat hard on the cells. So inducing a double-stranded break, for instance, is really hard on the cells. Has some kind of negative properties. So a similar system of targeting a particular region of the genome is something we call epigenome engineering or epigenome editing, where instead of hooking up a nuclease to a targeting protein, we can hook up a chromatin modifier or a transcription factor effector and induce a change in gene expression. So you can imagine like if the cell's expressing this protein that is causing problems, you could then deliver something that's going to repress the expression of that protein and cure that cell of that problem. So that's what kind of like my bread and butter has been for a long time. I worked on that type of technology at a company called Sangamo, and then also did some of that work in my postdoc, modifying T cells, which are a really important product for cell therapies, which is an exciting space in the biomedical field. And Tune was really started as like a platform for epigenome engineering, basically interested in targeting a number of diseases using that type of approach. And the backstory there is that we haven't had as much clinical precedence for this type of technology as we have for things like gene editing. But I'm really hopeful that Tune will be the company to be able to tackle this problem and get these types of reagents into the clinic and start to see some results for patients. So.
Yeah, that's super cool. The name tune makes sense, in terms of fine tuning the expressions and things instead of just for people that aren't familiar. A nuclease is something that actually cuts. It's an enzyme that cuts your DNA and actually breaks it, right? So you can engineer that to basically insert DNA, to remove DNA, to do different things. But what Jocelyn's is, she's not messing with the actual DNA itself, just the pieces of proteins that are around it that sort of control which genes are turned on and turned off, when and how much. And things like that.
Yeah.
Yeah.
Yeah, and if people are interested in learning more, our website's tunetx.com. Shout out to Glenn Murphy, who just revamped our whole website, and there's a bunch of really cool visuals for how this technology works on the website now. So encourage people to go check it out.
Nice. Awesome. Cool. And so T cells you mentioned previously, was that CAR T cells? I'm just curious.
Yeah, I mean the epi editing aspect can be applied to any cell product essentially. Like you could think of it as an add-on to the CAR T cell. So CAR T cells are T cells that have been engineered to express this CAR protein that allows them to target a specific type of cancer cell essentially. It works quite well for certain blood cancers, but doesn't work as well for some solid tumor cancers. And many companies are working on improving that system and trying to target more diseases with it. But yeah, I think one of the challenges in the solid tumor space is that the cells get into the tumor, and we can see that. We can, use pathology and image that the cells have gotten into the tumor, but then they tend to become exhausted. The cells get tired, and so hopefully, certain types of modifications to those cells allow them to continue to kill cancer cells and overcome that state.
Yeah, I don't know much about it. I remember reading about it, learning a little bit about it when I was in grad school. Considering either looking for postdocs or going into tech, into a biotech company. But I haven't kept up with it, so that's great. I'm sure it's advanced tremendously.
Yeah, it's an exciting space, and I mean, when I was at Lyell, which is an immunopharma company focused on these types of cells, we often talked about cell therapy as this third wave of medicine. If you think about the first wave being small molecule drugs, the second wave being biologics, things like proteins, antibodies, and the third wave being cells as products. It's a pretty interesting shift, and there are some caveats to that. It's a very expensive process the way that most companies do it right now, but there's so many people focused on making this better and making it more accessible, and I'm very hopeful for the field.
Yeah, definitely. I mean, something that can target cancer like that, and even just cell therapies in general, it really is that next building block. And it reminded me of the recent interview of Michael Levin on Lex Friedman, where he's talking about engineering more complex things like materials out of cells and it's living substrate. Reminds me of what my undergrad advisor told me. I was working in an organic chemistry lab, and I was telling her that I was going to grad school of biology. She's like, "Good luck, I gave up on biology because those things are literally alive. Your experiment has a mind of its own." So
yeah, that was a really illuminating interview. I haven't listened. I think I listened to the first hour. But yeah, it's so interesting. What they're working on, like, what do they call them? Xenobots?
Right. Yeah.
Which is
Xenobots.
Interesting.
These little cells and it's crazy how they even form. So he was doing basically during embryo dissection with Xenopus, it would sometimes shed off these chunks of the not the evidence, but the blanking on the word, that outer layer of cells. Right, they're essentially they're to become stem cells, but they're not differentiated and developed yet. And then breaking them off at a certain stage, they would sort of lose these developmental cues that would normally induce them to continue differentiating. They just break into these little aggregates, of various sizes and literally start moving around the dish and can like navigate a maze. You know,
Yeah.
it's crazy. Yeah.
Yeah, it's an interesting kind of engineering mechanism to understand biology. I find that very interesting. I loved his point too about like organism awareness. Like I guess it makes sense, but it didn't really click for me until I heard him talk about that, of like an organism becoming self-aware of its environment and where it ends and the environment begins, right? Which is really interesting. I have a 3-month-old, and like watching her go through that process of like, oh, this is my hand, you know. It's a fascinating aspect of biology, so pretty interesting stuff.
Seriously, yeah. Well, we could go down the rabbit hole about consciousness, but I know I'm not an expert in that, so I think we'll save it for another day.
Yeah, definitely. Like, I mean, for especially for anyone who's worked in the neuroscience field, some really interesting questions there. I actually interviewed this researcher, Kanika Rajan, and kind of grilled her about consciousness and like all of her ways of thinking about that in an interview for Lady Scientist podcast. So recommend people check that out if you want to get into some like interesting questions on that, and how like can we even really measure consciousness. Like she studies how decisions are made in the brain, and yeah, very interesting discussion. So
Yeah, that's awesome. I definitely I'll put that in the link in the show notes. I'm gonna send you a link to that.
yeah, I will.
Yeah, awesome, cool. And that kind of like brings up this basic aspect of science. Like, hey, we can't just measuring things about the world. Right, we're so limited in what we can know, and we can even logically, like epistemologically, we're dealing with the philosophical aspect of you know just even identifying ourselves, right? And in science, kind of people take that for granted, and like the scientific method, testing a hypothesis, having a prediction, right? A testable prediction, an explanation world. Um, People sort of lose track a lot about that. That's something that was really the main focus of grad school for me, like developing a hypothesis, developing an experiment. And it's just so fundamental and so basic, and it just gets lost because we kind of forget about it. I feel like people forget about it, right? Scientists, maybe practicing scientists, hopefully not too much, but it's really not part of common parlance and talk about science, right? People say, "Oh, I have a theory." And that's a theory is not an idea. A theory is a collection of facts, about the world. And so just the way we'll talk about science, I don't know. Like that's partially why I'm passionate about science communications and having podcasts like this and starting to try and delve into maybe more complex topics, but hopefully break them down into a simple way.
Like the porous nature of science, is that what you're getting at?
Interesting. What do you mean? Can you say more about that?
I guess what came to mind as you were describing it is that the kind of basic unit of science is a question, like a hypothesis or a question, and that then that is like explored or evaluated, and it's not it takes time for it to become like a fact, right? Like a lot of science is more free flowing than I think people outside of it necessarily can comprehend. Maybe that's not the exact way to describe it, but like it's like the actual practice of being a scientist and exploring questions is much more vague, I guess, than like a textbook of knowledge, right? It's more like a conversation where you're questioning things, and like oftentimes within a given field, like let's take genetics or gene regulation or something, people are presenting their data as they understand it, and it's through this cross communication with other peers in your field that things become clear and like consensus is derived, if that makes sense.
Right. Yeah. No. And that was a good transition between like the process of doing science to the process of scientific knowledge, right? How does scientific knowledge become a thing?
Mm-hmm.
And that's actually a good segue into kind of DeSci things again, because in DeSci and what you're doing with the wiki, mapping knowledge, there are a couple cool DeSci projects that are doing things like that. A couple that talked yesterday, A for tech trees, for example, for what Foresight doing, mapping the relationships of different organizations and what they're working on, the problems working on, how they get to a ultimate solution. And things like a knowledge graph, right? How do we the citations, like what DeSci Labs is doing in terms of things like that.
Super exciting.
Practice.
Yeah, I find the DeSci Labs nodes project, I forget what name they're using now, but I find that to be one of the really exciting products, if you will, that's coming out of DeSci right now. Because you think about like our global understanding of the scientific literature, and how siloed a lot of that is, and how poorly we can interact with that body of knowledge. And then you start to apply some of these technological advances to essentially being able to do function calls on particular pieces of information in papers. That can have a huge value add to, our communal interaction with the literature. And I've been kind of doing some research on different people working on knowledge graphs for science. There's a number of cool projects in that space. I know Protocol Labs has been involved with this discourse graph through Rome Research that a couple people are using to generate knowledge graphs for things like hypotheses or questions, or even companies that are working on particular topics. So that's something this group Deep Science Ventures is working on. They have what they call outcomes graphs utilizing that tool. So it's pretty exciting space, and definitely something we were excited about for LabDAO early on was being able to have knowledge graphs for biomedical research that happens within biotech companies. That's normally a very closed source. And if we were able to incentivize people to share more information, even about like the type of experiments that they're doing. So like every company in a certain area might have to do a mouse study, and being able to share things like the number of mice they use per arm, the different doses they use, that could add a lot of value to the system. So that's something that we continue to explore at LabDAO.
Yeah, I'll get into LabDAO too. But kind of like wrapping up this really esoteric segment that we're kind of going through. The doing of science is almost kind of it's being more formalized, I guess, as we're applying this tech. We're kind of going from, we're helping sort of facilitate the formalization of like doing science, which is very creative, right? It's this creative process of thinking about a problem and trying to question it and poke and probe, and then actually doing it itself, right? I know people are talking about publishing methods before you do them, which is appropriate in certain contexts, especially like human studies, clinical trials, and things like that. A lot of the time in the lab, especially when you're working with model organisms, it is much more free-flowing, fast-paced. A lot of times you're doing something in the lab and you may write out the protocol beforehand, but it's not until you're doing it that you realize, oh, I need to change this and this and this, and do something a little bit differently to actually make it practical to accomplish this protocol or whatever you're trying to do. So it can be very creative and artistic in terms of how science is actually done. Obviously it gets formalized and published in a paper, but even applying some of this type of knowledge to graphs and tech trees and things, and function call citations especially. Going through that process of getting data onto the DeSci lab codes really made me stop and think, oh, like my manuscript is nowhere near ready for this right now. Like, I need to go back and add a bunch of things, or get my data, my code together, because it needs to be published in a certain way. So I think that will have a feedback effect on the doing of science, right?
Totally.
Yeah. So wrapping that esoteric portion, let's talk maybe a little bit about, we mentioned the DeSci Wiki and LabDAO. So maybe you're involved in a lot of things in DeSci. Maybe give us a little background about how you got involved there, how you ended up doing the Wiki and LabDAO.
Yeah. So really started down this like Web3 science rabbit hole through conversations around scientific publishing, in fact, and how the tokenization of scientific publishing might improve, the process of sharing information in the system, incentivizing peer reviewers, things like that, and just like it became such an interesting thought exercise. And then I started talking to some of the folks in the space early on, VitaDAO. There's, a number of people who are like applying these concepts of decentralization, or, spinning up DAOs to focus on particular scientific problems. And so I was talking to all these people in the space really for my own like research and trying to understand the space and what was being worked on, and started thinking like, wow, every conversation I'm having is so inspiring and exciting. Like the way people are thinking about these things and kind of the like on the fly innovation that was happening, that I wanted to share these conversations with others. So as part of our project Ultra Rare, I started a show Ultra Rare, the podcast, where I interviewed some of the thought leaders in the space and put together the wiki as a way of like kind of like organizing the people that I was talking to and what they were working on and all the links, and opened it up, to the community, and just started expanding on it as I talked to more and more people. And it really became kind of a resource for folks trying to understand the space. And, I broke it down into different categories of like what people were focused on. So, decentralized biotech was a big theme for me. I was interested in how decentralized biotech was being applied and all of the key players in that space. Scientific publishing, of course. I also have a section with books that a lot of people recommend in the space, podcasts, articles, and we've even introduced like entirely new categories as time went on, like protocols that people are developing. So things like the Ocean Protocol or Bakalhau, which I think they're at the conference you're at. And ecosystems. So there's people who are like trying to build entirely new scientific ecosystems that are tackling multiple problems within science. So yeah, those are kind of the sections in the wiki. Recommend people, add to it if your project is missing. So it's a Google Doc. It's a living document that anyone can comment on with their links and their project that they want to have added. And I go in there maybe once a week and work on integrating those comments.
Nice. Yeah, it's a fantastic resource. So if anybody hasn't seen that yet, it's a great document. Go ahead and add stuff to it. If you're listening to this and you want to get your product in there, and then you have this awesome graphic too that kind of summarizes it all, that little like landscape ecosystem map.
Yeah. So that was put together by our Ultra Rare team, myself and Danny Berge, who's our lead artist. She's done a lot of really awesome scientific visualizations too. She did the artwork that goes with some of Molecule's IP NFTs. So if you've seen some of their IP NFT sales, they have this beautiful graphic of like actually what's happening to the cell as a result of that drug. Those are all done by Danny, very talented artist. So, and we also worked with a graphic designer to put that together. And yeah, it seems like it's been a useful resource to the community. We try to update it like, every few months or so. But it is a lot of work to collect all that information and logos and everything. And yeah, it's a good way to check out what's happening in the space and see what other DAOs or communities are out there. There are a lot of like parallel ideas and projects in the space, and I'm hopeful that some people will find ways to join forces and really accomplish the goals that they're going after.
Yeah, definitely. I keep my eye on it for sure. So thank you for doing that. So you mentioned ultra rare while we were in there. You gave a brief description. But maybe what's been the highlights of doing ultra rare?
Yeah, ultra rare started as this like little collective group of friends. We have a Slack, and our mission was like, what can we accomplish at the intersection of like science, art, communication, and philanthropy. And so long-term goal is to have an NFT collection that raises money for some of the research that rare disease families have to carry out, which can be very expensive. And the current process for these families is they go through a kind of fundraising for their nonprofits so that they can then carry out these studies that cost hundreds of thousands of dollars usually, and identify if there's any drugs. Oftentimes they're looking at drug repurposing. So they're taking panels of drugs that are already approved that they can acquire that might have an overlapping mechanism for the rare disease that they're trying to alleviate. So that was the initial goal, and until we're ready to launch NFTs, we've just been supporting the community through a lot of this messaging, graphic design, art, and communication. We did a POEP design for this drug repurposing course that this guy Sanath puts on for other families. Since he's gone through this process himself, he's now teaching others how to do it. So things like that we love supporting in whatever way we can. And yeah, it's fun. And we of course support the wiki and the DeSci landscape as well.
Yeah, that's huge. I mean, decentralized science and funding for rare diseases. Name, ultra rare. It's kind of it's got that vibe to it.
Yeah, it's kind of a play on super rare, if people are familiar with that. But ultra rare diseases, of course, are that's also a descriptor for these super rare disorders that there might only be a handful of patients, and therefore they're kind of missed by canonical biotech companies because there's not a big enough patient population to justify researching or doing the drug development for that disorder. So they're a really important use case for this type of decentralized science, and similar to the democratization of biotech, being able to share the information it takes to do that drug development so that these families can do their own drug development, which is exciting. It's really cool to see. I mean, even compared to a year ago, how many more people are building in public and sharing the types of information that others might need to advance their own drug discovery journey.
That's awesome. You mean in terms of biotech and things like that?
Yeah.
Outside of DeSci, traditional companies? Wow.
Yeah, I mean, I just feel like it's become a more common conversation, and there's a lot of people that are passionate about sharing what they've learned and the knowledge it takes. And of course these rare disease communities were already the hub for this type of knowledge share before this, but we see more and more VCs even who are investing in biotech companies doing this type of knowledge share. So there's been a big movement, the founder-led biotech movement, that was really kind of headed up by Petrie Pillar and Tony Kalisa, and a number of other people in the space. And they actually just are wrapping up their second founder-led summit. And as part of that, they've put out an ecosystem map for biotech, founder-led biotech, and it has, you know, podcasts, it has blogs, it has classes, it has all kinds of resources on this webpage, fully interactive. And if they're missing anything, you can comment to add it. And yeah, it's just a great resource, I think, for people who want to work in this space. And it's a hard space to work in, you know. Biotech is hard, so.
Biotech is definitely hard, and it's really hard to found a biotech company. That's from firsthand experience.
Yep.
Yeah, and hopefully that DeSci, that's definitely something, you know, just broad themes of DeSci is right, funding, access to resources, and support, and infrastructure is going to be one of those big ones for sure. Right, how do early stage scientists, people are talking about tech transfer right now from universities and sort of decentralizing that, getting tech out, but you know, even having spaces for scientists to work, you know, in the sense of being able to generate IP that doesn't need to be transferred, right? They're part of this, for DAO, for example. So I think we'll get there.
Yeah, there, you know, along those lines, there have been some really great resources shared in the last year as well for spinning out of universities. So there's this, I think the website's called spinout.fyi, put together by a guy named Nathan, that has been aggregating data on spin out deals. And this really helps founders understand that process more. And again, encourage people to add to that. I think it's a kind of open living document and resource. Currently has, like, you know, data from 150 different universities and, you know, how those deals work. But yeah, traditionally tech transfer hasn't been as streamlined as we would like. Like, I mean, you take a university like University of Washington, for instance, that generates a ton of novel IP on a daily basis, and yet traditionally hasn't been as easy to spin out companies as you might like it to be. And I think a lot of people are trying to improve that, but information can be valuable in that regard, and helping people spin things out. And then another way to get around this is people are starting places like Arcadia Institute, which is kind of this experiment in a new translation model where researchers are doing basic science, but then as things develop that might have applicability as a product or a drug, they can incubate and de-risk that at the institute, and then help those companies form. And there are other examples of those types of institutes, like the Institute for Systems Biology, led by Lee Hood, has always traditionally had a lot of spin out companies. There's also, like, you know, the IPD within University of Washington, led by David Baker, again, has this very enriched culture for spinning out companies. And I think, you know, a lot of it comes down to, like, that leadership, but also the, like, parallelization and helping each other with the information you need to start those companies. So I'm excited to see more people experimenting with that model, and very hopeful for it allowing for greater translation of things like basic research.
Yeah, definitely. And I have to remind you to send me those links for those three you just mentioned. And I think you'd already sent me Arcadia, so I think if you had commented on a post I made on Twitter about that, because so kind of what I'm trying to do within Real Lab. You know, we've had this molecular genomics laboratory down in Santa Barbara, and it operated as a diagnostic lab for years. You know, biotech, commercial biotech, basically, and that was really tough. You know, I'm a researcher. I always said during grad school, there's no way I'm going to run a diagnostic lab. Guess what I started doing immediately after grad school was starting a diagnostic lab and a little biotech company. But that was good. That was a great learning experience, but it was really hard business model. So we ended up closing that down earlier this year, but I still have the space. The lab is just sitting empty and unused. So kind of retooled a little bit, organized it a little more. And so now we're going to be renting out the space as a co-working space, and tokenize it. So basically the participants, the people that are renting the space, can run workshops, boot camps, either in real life boot camps or virtual courses, and basically get the majority of the profit from that, but sort of promote their work and be able to fund their own research, kind of like through teaching, kind of like a lot of people did in grad school, TAing their way through grad school, but offering that as a way to by tokenizing it and rewarding them for contributing to that effort and producing content, they will get that ownership and governance in the system, and be able to same thing for the IP producers. That's kind of the goal there, is to build that digital collective, that cooperative around this co-working space and producing content.
Love it.
Yeah.
That's super exciting. Yeah, I saw you promoting the space, and I mean, lab space tends to be pretty limited these days. So hopefully, people are interested in utilizing the space. And I think, like for instance, this company Perlara, they have a pop-up lab at this SF Biolabs co-working space, and have some great experiments going there. And yeah, I think the reality of science is we need physical spaces to do a lot of the work. It's not a fully computational operation, at least not right now. So yeah, it's awesome that you're like providing that to the community, and I'm excited to see what comes out of it.
Yeah, thank you. Me too. I really hope that I meet some cool people who start doing cool work, right? I always loved teaching, and I wanted to get back into maybe do some sort of virtual course after running a business. I was like, oh, maybe I'll do something in entrepreneurship, and I can just do all of it at once. So
yeah, that's rad.
But we're right down the street from UCSB. So I did, I was first starting my business. I was still in grad school. That led to the business plan competition on campus. They have an annual thing, and they do have a little incubator space there on campus, but it's just a small office in the basement of this one building. There is some lab space, equipment that you can use if you're not affiliated with the campus, if you've graduated or whatever and starting a business and you want to access, say, a sequencer or PCR machine or clean room or something like that, you can do that. But it's a little more difficult. It's definitely not financially affordable for a lot of people, and so we're really trying to create more flexible, accessible space.
Yeah, I'm a big fan. I mean, Alexandria and some of these other biotech real estate companies are also generating additional space for people to rent out, like essentially single room suites. And I think that type of resource is really valuable for companies that are just getting started. And just to give people a sense of the numbers, like I think, your lease might be something on the order of like $10,000 a month. So yeah, it's an expensive business to be in, and any way we can help each other through these like co-working, co-lab spaces, I think can add a lot of value to companies that are just getting started.
Yeah, definitely. Right. Now, that was the whole business model for the diagnostic lab, was that we do rapid PCR diagnostics, a lot for focus on cannabis. So we do it for cannabis growers, and a lot of these growers, they would want to set up their own lab on site. There was other companies selling on-site testing kits and things like that, but it didn't make sense, for them to build their own lab, staff their own lab, maintain it, right, buy all these really reagents and things, and then waste a bunch because they're not doing it properly or whatever, and they're not getting good results. So, we would pitch, okay, outsource all that to us. We'll run it, send us a little sample. And that's kind of, I think, how I'm approaching this for DeSci projects or for early stage biotech, right? Like, not a lot of people need a full-time lab. Don't need a whole lab, right? They need short-term access to get an experiment done, to generate some data. And then they take it online, right, do their analysis, do whatever they got to publish it. So.
Nice.
Yeah. Well, enough about me. So we've got a couple more things I want to get into, and we mentioned funding a little bit previously, but you recently published an article, or co-published an article on Patty McCormick's website, Gassing the Miracle Machine.
Yeah.
So, yeah, fantastic.
Thank
Maybe
you.
give a little bit of summary and what's your what was your thesis with that article, and what's in terms of funding for science and thesis.
Yeah, for sure. Shout out to Elliot Hirschberg, who writes this awesome blog, Century of Biology, and he's been writing for Packy on Not Boring for a little while, and invited me to co-author this piece with him, where we really explored, kind of the traditional scientific state of funding, and then contrasted that with some of these new experiments that are happening both like in the Web3 space, but also some of these new institutes that are forming that I mentioned. And just some of the new kind of like funding models that are being explored with things like fast grants. So the kind of gist of the article, we go through how NIH funding works and the backstory there, as far as like where most of the funding goes to these PI centric university labs, and over time that has become pretty bureaucratic and consensus driven process. So like the traditional NIH grant that most PIs have to go after are these R01 grants, and they take about a year to be evaluated. They're evaluated by these big committees that suck up a lot of people's time. And, by some estimates, like these researchers are spending, 40% or more of their time working on just writing these grants, and not really focused as much on like doing the science. And as they get further in their careers, it becomes a lot more like administrative and managerial. So there's a whole kind of structure to the system that I think has become, in my eyes, somewhat of like the predominant model of basic research and how it's done. And the ultimate goal and thing to come out of that type of research is also papers that are published in these traditional, what I call big journals, that, it's gearing people to a specific type of research. And I think my argument is that, like, when you have one very specific modality for doing research, then you get a very specific result and output. And if we have a diversity of models, if we have a diversity of how things are getting funded and who's getting funded and at what stage of their career and within different types of institutes, that we might also get a more diverse output of results, and therefore scientific progress. And so that's kind of like my working thesis, is like, well, why not experiment with the status quo and the existing systems that we're operating in, and see if there's something that comes out of it. Like, what if we get more more cures for rare diseases, or more leaps in understanding for different types of technology or different scientific questions. So yeah, that's kind of the idea there. And I get into really a lot of the like exciting pioneers in these spaces, across funding, across DeSci, and also across these new independent institutes that are forming in the article. So I really encourage people to check it out. And it ended up being I think it ended up being over 7,000 words. So it's a little bit long, but hopefully people get something out of it.
Yeah, no, great read. And it's so true. It brings up the question, honestly, of what sort of, hopefully there's not more strings being attached to it, because I know when you get a grant from the NIH, something, for example, there's strings attached in terms of how things can be published, and requirements, obviously, to publish and get work out there. But yeah, some money comes with strings. So hopefully there aren't more strings with more, I guess, gas tanks to fill.
Yeah, there was a really interesting article that we read as part of our research for this piece that was published by the folks who put together this FAST grants program during the pandemic. And they described some of their learnings. So this was in Future by A16Z, Patrick Sue, Tyler Cowan, and Collison all co-authored this piece. And they talked about like the rigidity of funding from the NIH and how basically like once you get a grant to do a specific study, let's say, you're kind of locked into like using that funding to do that thing. And so there's like less flexibility than you would think. And in this survey, they surveyed, people asking for this type of money, and a lot of people said, yeah, we'd love to be able to do some other things and like have flexibility and do what I call like blue sky research. And it's, they make the interesting point of like, it's almost like the bureaucracy is such that we're not even trusting the innovators and the scientists themselves to like make these types of decisions of like what they're going to study. So yeah, it's kind of an interesting conundrum we've gotten into, and one that I think, some of these organizations like VitaDAO are, kind of part of their thesis is like, oh, we're going to fund some different projects and we'll see if that's true or not. But yeah, I'm excited to see people experiment with this new source of capital in the form of crypto and maybe fund some alternative projects. And I think the recent DeSci Gitcoin round was really exciting for this. And I again talk about it in the article, but super excited to see a lot of these grassroots science projects get off the ground as a result of Gitcoin.
Yeah, no, Gitcoin's fantastic. It's good to see Gitcoin continue to grow, and then the DeSci round was huge. So many projects. For me, there's so much value in meeting other projects, meeting other people. Right, there's tons of Twitter space almost every day of different founders or contributors to these different projects hopping on. I met a ton of people during that time. And there's just so much value that comes out of that, and especially if you consider the amount, there was a $500,000 matching round. I think more than that was donated for sure. And in terms of science, like that in total is like in one confocal microscope, right? Like DeSci is like the biggest bargain revolution in history, I think.
The biggest what?
Bargain revolution in history. Like it's the amount of progress being made in DeSci and projects that are getting things off the ground with funding. It's incredible.
Yeah, no, I love that. That's a good way of describing it.
Yeah. And so hopefully more products will continue to work together, because that's really the network effect of quadratic funding, right? Being able to donate $1 to 50 projects, and then have that $50 be matched by $4,000, something crazy, whatever it was, depending on the projects. The matching works really well, and it's all about that community and putting the kind of back to what we were talking about earlier, putting the users of science in the driver's seat. You can pick what you want to fund. You can show up and talk to the people in Discord. And when projects work together and even fund each other, it creates, Kevin Nowacki, the founder of Coin, called it a jet engine of funding, right? Because it's like turbocharging things. Projects are collaborating instead of competing. We're all on spaces together, talking about our projects together, like being like, oh, that's super cool, let's work together here, instead of trying to compete super hardcore against each other and saying, oh, I'm not going to talk about this with you or this, I love the collaborative aspect and how that's shifting everything.
Yeah, it's, I think, so important if you're trying to do science to recognize that it doesn't happen in a vacuum. Like there is, it's very hard to make progress like on an island, and you really have to lean on your community. And that's part of why universities work, right? Like having departments with your peers down the hall where you can go and bug them with questions, and having, you know, beer hour on Fridays where you brainstorm ideas with your colleagues. Like, a lot of like progress happens in those like interactions, right? And so, you know, one of the exciting things about DeSci is that we're now building some of these, you know, interaction spaces online so that anyone can access that and anyone can participate in those conversations and learn from each other and come up with ideas, which is pretty exciting, you know, because most of these like university halls or beer hours, like there's only what, like so many of them, and you have to like live in that city. You have to cater to that career path to like be able to access that community, right? So now, you know, we're breaking down the barriers to that and more people can be a part of that conversation.
They can be a part of the conversation and really get like hands-on to the problem itself, you know. And yeah, with this funding, things like Bitcoin puts also the power back into the hands of scientists, right? Because there's no strings for that. A lot of things that I love about DAOs is the modularity, the focus on putting the people that are closest to the problem in the decision-making seat, and giving them the power to do that. And yeah, kind of back to what we were just talking about with funding science.
Yeah.
Well, awesome. I was, maybe we'll mention just your blog here at the end. You also have a newsletter actually on Substack Motif.
Yeah.
So you've got even more writing.
Yeah, I just started that actually somewhat specifically for this article I wrote about the changing landscape of scientific publishing, because I had pitched this article to a number of outlets and just like wasn't landing anywhere. And I was like, you know, I just got to get this article out because I'd been researching all these people working on this problem. You know, there's a couple themes that had come out of my conversations with people. So improving the peer review process. A lot of people are interested in this idea of micro publications. So like most of the literature is published in the form of these long form articles that take on average about a year to put together and maybe another year to publish, sometimes more than that. Like I have a paper that we still haven't gotten out from when I was in graduate school, and I graduated like 5 years ago. So it can be a pretty lengthy process to like share that information with others. And so some people are exploring the idea of these micro publications, like single figure papers, which I think is pretty cool. So I cover some of the like progress in that space in this article, and I'll be doing some additional deep dives on my blog there, on Substack. And yeah, I'm excited to like share more of my research in this space with others, and encourage folks to comment and let me know what you're interested in. If there's particular topics, you know, feel free to shoot me a DM on Twitter. DMs are open, but unfortunately, I get a lot of spam as a result of like all these NFT airdrop blah blah blah. Just wish I could get rid of that. But yeah.
Yeah, for a while they were going into that like not the main folder for DMs. I get a ton of them. But
yeah.
Hopefully we'll see what Elon does about that. I don't know. A lot of
Yeah.
hubbub around all this.
I'm also bummed because like it's just an unfortunate aspect of like the Web3 space. Is that like this type of spam has become so much worse. But Telegram too now. Like Telegram, like I'm getting added to all these like chats on Telegram, and luckily reporting spam on Telegram is easier than reporting it on Twitter. But it's still like a very unfortunate aspect of the space, I think.
Yeah, for sure. I know. I used to have my Telegram settings closed, and I opened it up because somebody like commented on it. And I was like, why did they do that? Why did they do that?
Yeah.
But I'm glad I got through
hopefully
you. I'm glad I was able to get you on the podcast today. Yeah, sorry for cutting you off.
Oh no, go ahead. Yeah. Thanks so much for inviting me, and I'm excited for DeSci.Party and to see who else you have on. So
Absolutely. well, check out Jocelyn's work, everybody. Again, senior scientist at Tune Therapeutics, host of the Lady Scientist podcast, creator of the DeSci Wiki, writer of Gat in the Miracle, co-author of Gas in the Miracle Machine, writer at Motif, among many, many other awesome things. So thank you again, Jocelynn. It's been a fantastic conversation. I really appreciate it.
Yeah, thanks so much, Elijah. I'm super pumped for all the work you're doing with the In Real Lab, and I can't wait to hear like what comes out of this co-working space and all the work you're doing in the DeSci space with MedDAO and all kinds of fun stuff. So I'm excited to follow along.
Likewise, yeah, let's stay in touch for sure. All right, we've got hackers
Awesome.
coming in 2 hours, so
Okay. All right,
Okay.
have fun.